and welcome to the Beyond Biotech podcast number 95. I'm Jim Cornell and this is the weekly podcast from Labiotech. This week we will find out all about Antisense oligonucleotides, which fortunately is a lot easier to say as ASOs. This week we had a chat with René Ruckert, Chief Operating Officer at Isarna Therapeutics, who explains transforming growth factor beta, or beta, depending on which side of the Atlantic you are, and antisense oligonucleotides, and their role in tackling disease, including Isarna's own work on wet age-related macular degeneration and diabetic macular edema. So I wonder if first you could tell me something about the company.
Yeah, so I actually worked for Isarna Therapeutics for 6 years already. I started in 2018 as the Chief Medical Officer. Then a year later, we could get a real expert in the field we're working in. So Professor Marion Monk is a retina specialist, and so she took over the CMO position, and I do COO, so all the operational stuff since then for Isarna. Isarna itself is a company based just out of Munich. It was founded in 1998 already, very early, so way before my time. There were many changes on size and location and people, so none of the founders is associated anymore. And kept on working and surviving for this almost 25 years, which is for a biotech startup a very long time. Part of this survival strategy is obviously investment by MIG investment in Munich, and they are still the key investor in Isarna Therapeutics. The company was called Antisense Pharma actually in the beginning. So the idea was really Antisense more or less from the beginning, but it was rebranded in 2013 as Isarna Pharma then. Isarna actually is based on a river that runs to Munich. So it's the Isar. Isarna is now a virtual company, more or less. Yeah, we have a handful of consultants. I'm one of them. We are spread over Europe and doing pretty good since many years already. So I think that is a business model that is cool.
Maybe we could start if you could tell me a little bit about transforming growth factor beta and what the relevance is in diseases and disease treatment.
Yeah, I'd like to. So transforming growth factor is a very complex kind of family of growth factors of proteins. It's called a superfamily, TGF-beta superfamily, which is based on 4 different groups that 23 different genes that coding for members of this group. There are very interesting, very important proteins that are part of the members, bone morphogenic proteins, so BMPs. They are really essential for growth and neonatal development. TGF-beta is relevant for it. But TGF-beta itself has different isoforms, so TGF-beta 1, 2, 3. I will later talk a bit more on TGF-beta 2, which is what we are working on and trying to bring to clinic. But TGF-beta itself has a pleiotropic cytokine or pleiotropic cytokines that are involved in many different activities in the immune system and also in the morphologic activity in the body. The relevance here is also particular for our drug that we are developing is that TGF-beta is a key driver of fibrosis. Fibrosis is a process that is involved in many different pathologies. We are talking on eye pathology or ocular pathology, I think in a minute. But fibrosis is a key driver of pathology in the lung. So pulmonary fibrosis, lung fibrosis is very severe disease. I used to be a doctor in pulmonary medicine, a medical doctor in pulmonary medicine in clinic. And pulmonary fibrosis is something it's very hard to treat. And TGF-beta is one of the drivers for this. The other I think most important part that TGF-beta plays in pathology is also liver fibrosis. Yeah, there are many different reasons for this, but TGF-beta is a key driver of this fibrosis, and I think that is also a part where this TGF-beta is under heavy investigation for different preclinical and clinical studies.
Why is TGF-beta a good target?
Yeah, well, it is based on this activity that I just mentioned. It's a good target, in particular for this fibrosis and or inhibition of fibrosis if you inhibit TGF-beta. And yeah, more about this in the ophthalmology in a minute. But TGF-beta is a cytokine that is really significantly elevated in fibrotic tissue in patients with fibrosis. TGF-beta is produced by many different cells and is released or is stored intracellularly, but it is a good target since you can really suppress fibrosis, has been shown in plenty, hundreds of publications, and therefore I think it's a good target for us in our niche or in our indication of ophthalmology, but in particular also in other indications. And I should mention probably for Isarna still, we have this one molecule, TGF-beta-2 Antisense, but we also studied TGF-beta-1, TGF-beta-3 Antisense, and there are many, many opportunities to go.
What are antisense oligonucleotides and how do they work?
Antisense, it's been around for more than 30, 40 years. So I think 78 was the first mentioning of an antisense molecule in the research literature. Since then, it has made a huge development in technology and also, like I say, druggability of this technology. Antisense is a form of a treatment or therapy that you can use that is based on, say, oligonucleotides or that target messenger RNA. Yeah, so RNA obviously is very important for the protein production and production of every signaling molecule, every molecule in the body. And this messenger RNA is obviously transcribed from DNA in the nucleus, and then it goes to the ribosome and is in the cell cytoplasm, and is transcribed into protein, say a protein. The antisense is a nucleotide that is designed particular as a target, to target a very specific messenger RNA to inhibit this messenger RNA. Yeah, the mRNA is then degraded or it's just the transcription is inhibited. So there are different mechanisms how it can work. The antisense itself is really designed out of nucleotides that match the genetic code of the target. Say TGF-beta has a particular genetic code, and our antisense has exactly as an antisense the code that is in RNA nucleotide that has a length of between 10 and 20 nucleotides that target this mRNA and prevents the development or the transcription of the protein. So really TGF-beta production is inhibited, and not like another disease or therapeutics is you just block the protein that is already produced, and it really targets very specifically one molecule. And that is an antisense. There are many modifications of antisenses. So to increase the stability, otherwise the pure antisense is what I used in the lab 30 years ago or 25 years ago. That was very, very unstable and would never survive an injection in a human body since it was degraded. And now there are plenty of different methods to stabilize it and to really also add other molecules to target antisense, particularly to the liver, if you add some sugars. So it's a very interesting molecule.
What is it useful for treating?
Basically everything. Being a bit biased as being in this antisense field, but there are currently more than 10 molecules on the market or drugs on the market that are based on antisense. I'm still surprised that there are not more, since, again, the treatment is very specific. It really targets one molecule could be responsible for one indication, for one disease. The main indications of approved treatments are often diseases. Yeah, so kind of neurological diseases, also infectious diseases are part of what has been approved. Can talk about this in a second. But there are now studies going on in very large, say more important, or also commercially more attractive indications like lipid overproduction and also cardiac pathologies. So that is where I think the trend is going to larger indications.
What advantages do ASOs have?
It's a very, very specific treatment. And let me point it to look at other treatments at disadvantages of, say, if you use an antibody, which is obviously the largest market, and I think from a commercial point of view, the most attractive market at the moment to have antibodies against this and that and many, many different proteins. So the so-called biologics, they target molecules that have already been produced by the cell and also already released by the cells. Yeah, so if you target interleukin-2 as an example, interleukin-2 will be produced and will be released. And if you want to block it, you use an antibody and try to catch what has already been produced and released and is in the circulation or has already bound to receptors and may have already have done some damage or some activity. In contrast, Antisense is really targeting, again, as mentioned, intracellularly the production of such a cytokine as interleukin-2 or, in our case, TGF-beta 2. So it's not released at all. It's really, you hit the target before it can do any damage or can even be produced. So that's a major advantage.
What are the different ways that ASOs can be delivered?
That has also changed and improved in the past. So, as I mentioned, when I used the Antisense technology to silence genes or the production of some factors in the lab 20, 25 years ago, it was a pure molecule, a pure nucleotide that was highly unstable. Now we can do targeted delivery, and we could deliver antisense in different ways. So back then in the lab, it was electroporation, so high voltage, dangerous things. Now you can inject antisense, and most of the therapies, I think all of the therapies that are approved at the moment, are injected in some way. So we are talking about subcutaneous injection, intravenous injection. For neurologic diseases, there is intrathecal, so-called injection. It's really into the spine, into the liquor. But based on new development, and I mentioned some modifications, there are new studies that really show also bioavailability via oral garbage. So really to do it as a tablet, and you can try therefore to make the therapy way easier, which again makes it easier to target larger populations. So for hypertension, it would make it very difficult if all hypertension patients need to come to the clinic to get an IV infusion. But if you can have just a tablet or pill with the antisense, obviously that makes it easier. That is cool stuff and cool developments.
Yeah. Is it still something that is developing and that you're still working on and learning more about ASOs?
Yes, for sure. I mean, we at Isarna, we don't do any basic research anymore. So we're a very small company. We are focusing on our clinical pipeline, the one drug that we have in phase 2 now. But there's plenty of research going on. I just checked today, there are more than 25,000 papers in PubMed on antisense oligonucleotides, which is, I think it's more than I expected. Again, the technology or the idea is around since 78. It took quite a while before the first drug was approved. And again, compared to antibodies, there are probably hundreds or so approved for different therapies, a bit more than 10 antisense therapies approved and on the market for very few patients and often indication. It's still a surprise to me why not more companies are working on this. And yeah, I think it's a promising approach. Also, I think the formulation is the hottest topic, saying, okay, how could you get the drug to become more stable, which is an important point of therapy and thinking and a doctor. There are recent data on new development, new stabilization methods that show that the antisense could be stable for many, many months. So we have with our antisense on TGF-beta in animal models, we have target engagement and suppression of TGF-beta seen out to 4 months, for more than 4 months with a single injection. So that is something that is obviously promising also for other indications. If you say you have a duration of effect for many, many months, even half a year, so you would need 2 treatments a year. And that's one of the developments that the antisense field is going to.
Are there any other challenges involved with ASOs?
Not too many. I would say it is a field that becomes more and more interesting, and RNA technology became famous, unfortunately, to say, just a few years ago with the COVID pandemic. And BioNTech or Pfizer and their vaccine that it was based on it was not Antisense, but it was an RNA-based drug. And so we have, I think, a push in this direction of technology where Antisense is really growing. There's another option also for antisense in oncology. As so said, we have TGF-beta. It also plays a role in oncology. We have very early preclinical data that has been generated many, many years ago. So we're not actively working on this. But you can also generate antisense against not only messenger RNA, which we know for decades already, but also against microRNA, for example. So mRNA, which is or non-coding RNA, where everyone believed until 5 years ago that that's just junk RNA, but it is not. There are very important molecules that regulate transcription and many, many biologic processes. So both the micro and the non-transcribing RNA are particularly important in cancer pathology. And therefore, I think it's a direction where it goes and where many, many options are.
Are there lots of companies working in the field right now?
It's getting more. There are a few companies, so particular Ionis is a company that is mostly focused really on antisense. I would say it's the market leader that has also drugs on the market. More larger companies are getting into this, and it's potential for growth, I would say.
Are they best used as a standalone treatment or in conjunction with others?
It depends. We are just running a clinical trial and seeing that it works in some patients as a monotherapy, as a single treatment, but also for other technologies. There are particular cancer therapy. There are many, many treatments that have to be done with 2 or 3 or even more different drugs to really get the cancer under control. The same is true here. You have few indications, few pathologies that are really based on one single growth factor. Yeah. And if you target it, you knock out the disease and you kind of cure or treat the patients. It's not too often in medicine. So often there's this multifactorial and you have to try therapy with different molecules. But having said this, again, there's a potential, particularly also of our drug that we just presented a few data 3 days ago at a huge ophthalmology meeting, the ARVO in Seattle. We presented the monotherapy and we've seen pretty good efficacy. So hope that will hold true in larger studies.
Speaking of your own work, could you tell me a bit about the pipeline that you have at Isarna?
It's small, it's short, but again, Isarna is a small company. We have one drug in clinical development that is called, no one remember, but it's called ISTH0036. We don't even have a name yet, so we are working on getting a real name to move forward, which is a necessity for clinical development. But it's a drug that it's targeted really on ophthalmology. I mentioned Isarna had some oncology projects in the past when it was still called Antisense Pharma. We have preclinical data on oncology. We have different molecules, Antisense, as I mentioned, also targeting TGF-beta 1, TGF-beta 3, which all could be relevant in fibrosis in oncology, but also in fibrosis. The idea is still pulmonary fibrosis, lung fibrosis, where you could even administer antisense as an inhalation, as a spray. So it's a pretty stable molecule if you do the right modification. The other thing still is liver fibrosis. Renal fibrosis is something we considered, but the pipeline itself is phase 2A molecule, this ISTH0036, where we mentioned just Professor Monk just the day before presented first data of our phase 2A study at the meeting, and that's our pipeline. The indications are huge. It's a blockbuster indication. It's age-related macular degeneration. It's AMD. The other indication we are treating in the study, or patients we are treating, are diabetic patients with so-called diabetic macular edema. Both are diseases of the retinas, back of the eye, which are very frequent in older people. So AMD is, as the name said, age-related, is very frequent in people that are older than 60, 70, or 80 years. And diabetic becomes a pandemic, more or less, already. There are more people getting obese, that it's a global health issue and a big concern that more and more people get diabetes. Associated with this is also the the ocular pathology that is called diabetic macular edema. We actually seen good efficacy in both, and we're just looking to partner. We're looking for investors or partners to continue the development of the drug. Seen a good safety profile. Also, what I mentioned before, safety of Antisense is very, very good, so has very few side effects.
All right. How does it work with the AMD? I mean, does it stop it or reverse it?
It's working differently to the gold standard. Currently, the gold standard in ophthalmology and in this AMD and DME are injections into the eye, so-called intravitreal injections, with drugs that are on the market for 15 years already. So the main players are Bayer, Novartis, Genentech. So Eylea is one of the drugs that I actually used to help to develop with Bayer. So I joined Bayer 2006 in the headquarters and managed the trials of this ophthalmic disease. They work with blocking antibodies, blocking VEGF. VEGF is vascular endothelial growth factor, and that is a cytokine that makes a lot of edema in the eye. The drugs working pretty good already that are on the market, and others, Lucentis or faricimab, Vabysmo, are on the market that all target the same molecule, and they work in the beginning very good against the edema. But all the patients develop after half a year, and later on they develop a fibrosis. And therefore the visual acuity, the vision goes down and goes down and goes down. Even if you treat with the gold standard, you can't do anything against it. And our drug is working pretty good first against the edema. It's probably not as efficient as the other gold standard. So you may, to your question before, we may need to have a combination therapy in a few selected patients. But in most of the patients, it works on its own with injection every other month. We could extend it to every 3 or 4 months, hopefully. And it really showed some benefit in suppressing the fibrosis and suppressing the development of this fibrosis, which is, again, the key concern, is the highest unmet medical need in this both indications, the AMD and DME. And our first data that we presented look very good. So looking forward to larger studies and then approval, hopefully.
What kind of timeline are you looking at for that? I mean, you're in phase 2A now?
As soon as possible. I mean, it's obviously, again, we are finalizing the phase 2A at the moment while we're speaking, and starting a phase 2B/3, whatever study, with the right partner. Yeah, so phase 3 studies in this indication, meaning hundreds of patients, thousands of patients. Yeah, so large programs in the past that Roche, Novartis, Bayer were doing had more than 2,000 patients. So we're talking about a lot of money, a lot of effort. But we are very optimistic. We are in partnering discussions. Also, right while I'm speaking, our CMO, Marion Monk, is at the meeting talking to companies. We hope to get it phase 3 then next year, end of next year. So phase 2B/3 kind of study, which would bring us to an approval in less than a handful of years.
Oh, that's good. What would success be for the ISTH 0036, and what can patients expect?
Yeah, if it is confirmed, what we see right now is that we have a long-term treatment that really improves the visual acuity, the vision of these older patients, and keeps it stable for many, many years. The current treatment, as said, are very effective in the beginning. They improve the vision very fast. So you can see an effect even after 24 hours in some of the patients if you look into the eye. But even if you treat use the most optimal treatment regimen, you use monthly injection, you see after a few years that most of the patients develop fibrosis and the vision goes down, which could lead to, after a few years of treatment, to really legal blindness. So patients are not blind in a sense of everything is dark, but they cannot read, they cannot see, they cannot drive, they cannot do anything else. You cannot focus anymore. with our drug, we hope to prevent this progression of fibrosis, which is causing this deterioration. So we hope that in a short time, relatively short time frame, we have a help for these patients, and obviously become a very successful drug on the market. We are more or less the only company that are working on this target. So also this is very fascinating and obviously helpful for Isarna. We have a product or a drug molecule that could be once a drug on the market that no one else is really working on. So most of the companies are working on improving the current gold standard therapies, the anti-VEGF therapies with drugs that have a longer duration. So you don't need to inject every month, but you can inject every other month or every 4 months. Some are working on gene therapies that actually reduces the need for injection even more, since you produce the anti-VEGF antibody in the eye, in the retina, which is an interesting target, but that would not address that unmet medical need that we are targeting on fibrosis. So again, we hope for huge potential for our drug that we are developing.
Is there anything else that you wanted to mention that we hadn't?
It's a very interesting field, and I would encourage more people to have a look in the potential of Antisense as a therapy that has very few side effects, that has a very targeted mode of action, and could really improve the therapy for many, many different diseases. And with this, again, Isarna, if we get a partner, we are also happy to develop the other molecules and more indication. So that is the wish for the future.
A really interesting and informative look at ASOs, or antisense oligonucleotides, and very positive news about the company's work on eye conditions, and of course the potential for much more. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you'll have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.